Today we're going to be learning about diversity metrics, which is one of the topics that I struggled with in biolab. So I have to explain it. Um, so in biology, species diversity refers to the variety and abundance of different species within a particular area or ecosystem, and it's a really important component of biodiversity. Sorry, it is a key component of biodiversity, is which encompasses the variety of life on Earth. So, with the variety of species and genetic diversity within species and the diversity of ecosystems. And species diversity is typically measured through two main components, which would be species richness and evenness. And species richness is the total number of different species present in a specific area or region. So, for example, if a jungle has 20 types of insects and 15 types of trees, then the richness of that would be 35. Okay, and then with evenness, it is the relative abundance or distribution of these individuals among different species in the community. So, for example, with species A and species B, if there is one individual in the group of species A and then three individuals within species B, then it would not be very even. However, if there were two of each, then it would be. So the combination of species richness and evenness provides a more comprehensive understanding of species diversity in a given area, and a highly diverse community may have a large number of species, which would be high richness, and a relatively even distribution of individuals, so high evenness. And species diversity is really important for several reasons. One of them being ecosystem stability. So, this is because diverse ecosystems are often more stable and resilient to environmental changes, because different species play unique roles in maintaining this ecological balance that is needed and in order to function. Another reason as to why it's really important is adaptation and evolution. And higher species diversity provides this because it provides a larger pool of genetic material, which gives more options for adaptive potential of a population. And this diversity is really essential for species to evolve and cope with how the environment changes. Um, Another reason is that its economic and ecological services. So many ecosystems with high diversity provide a lot of valuable services such as pollination, water purification, and pest control. And a loss of this diversity can impact these services, which is not great. Um, the last reason is conservation, and understanding and monitoring these species diversity is super important for conservation efforts because it aims to protect the variety of life on Earth, recognizing value of biodiversity and its importance to human well-being as well. And so scientists like to use various tools in order to measure biodiversity, such as the Shannon Index, which was mentioned in class. It's commonly used in biology to quantify the species diversity in a community, so it takes into account both the number of different species and the evenness of their abundances. So this provides a single numerical value which reflects the diversity of a community, considering both the varieties of species are present and the distribution of individuals among these species. Some of that is some key points that are important about the Shannon Diversity Index are that higher values in the Shannon Diversity Index indicates higher diversity, and this occurs when there are many species present and the distribution is more even. There and then the Shannon Index is also sensitive to rare species, so it considers the proportion of individuals for each species. And all in all, researchers use the Shannon Diversity Index to assess and compare the diversity of different ecological communities, ecosystems, or habitats, and it's a valuable tool for conservation efforts. Next, there is beta and alpha diversity, which are two components of biodiversity that are used to quantify and understand different aspects of species diversity within ecosystems. So, alpha diversity refers to the diversity of species within one particular habitat or ecosystem, and it is a measure of local diversity and is often expressed as the number of species or richness of a diversity index within a specific area. So it provides insights into the number and variety of species in a single location, and that helps researchers understand the ecological complexity and richness within one given habitat. So, for example, if you're studying the biodiversity, or not the diversity of plant species within a forest, the number of plant species present in that specific forest would be a measure of alpha diversity. However, on the other hand, with beta diversity, it measures the turnover or change in species composition between different habitats or locations. So it quantifies a degree of differentiation in species composition between two or more ecosystems. And the reason that biodiversity is important is because it helps scientists understand how different or similar various habitats are in terms of their species composition. So it provides information about the degree of heterogeneity, or similarity between ecosystems. So, for example, if you compare the plant species present in a forest, a grassland, and a wetland, then the differences in species composition among these three habitats would be a measure of beta diversity. So, in summary, alpha diversity focuses on the diversity of a species within a specific location or habitat, providing information about the variety and abundance of species in that particular area. And then beta diversity focuses on the differences in species composition between different habitats or locations, which helps researchers understand the turnover of or change in species as they move on to one ecosystem to another. So together, alpha and beta diversity contribute to a more comprehensive understanding of overall biodiversity patterns within and between ecosystems. And that is all for my subject of diversity metrics. Thank you for watching. Have a great day and subscribe also. And also leave a like.